So you get a ratio of 9 blues that are big A and big B, 3 whites that are big A, little b, and then you get 4 yellows. 3 of them are little a, big B in something, and 1 of them is little a, little b. But then all 4 of these will show up as being yellow. Okay, and so the ratio becomes 4:3:9. The step yellow comes out first, followed by the 3, followed by the 9. And that's the pathway in which they work. All right, so let's take another example of recessive epistasis, but that gives you a slightly different color and a different ratio. So, before we do that, let's take an example that you've probably seen, which is coat color in dogs, Labradors. Okay? And this is basically recessive epistasis. So labs can be black or yellow or brown. Right? So let's take a cross. Here is an example slightly different. You start with a true breeding black lab, cross it to a true breeding yellow lab. And I'm telling you that it's not true breeding in the sense that I just talked about, but it's true breeding such that it's little b, little b, little e, little e. Okay? And you get basically a black lab in the F1. And you cross a black heterozygous to a black heterozygous, and the pathway for coat color in labs is yellow, brown, black. So let's say you went to a breeder and you wanted a black lab. Right? It could be, you may want a true breeding black lab. He may give you a heterozygous black lab. It's entirely possible. The way to figure it out is to basically cross that lab to another black lab and ask what happens. If it's true breeding, you'll get a different ratio. If it's a heterozygous, you'll get a different ratio. Right? And that's how you basically figure it out. Okay, so when we do this cross, this heterozygous black lab, big B, little b, big E, little e, will produce 4 kinds of gametes. And if you were to cross these, you would get labs in the color 9 blacks, 3 browns, and 4 yellows. And based on this, you could draw out this part. Instantaneously, you could say, yellow is the first step, followed by brown, followed by black. And there are 2 genes, 2 enzymes that are converting yellow to brown. Yes? Any questions? Okay, so now let's do another cross. So this is known as epistasis, when the allele of one gene masks the expression of an allele of a second gene, or alters the expression of a second gene, the allele of a second gene. It can either mask it or alter it. That's known as epistasis. And it's basically being observed in this individual right here. The final individual. Okay, so this is known as recessive epistasis, where the homozygous little e gene allele masks the effect of the big B allele that's working downstream. And little e is epistatic to B, little e works upstream of E. It works before E. So or little r before B. So the E enzyme works before the B enzyme. So epistasis is just a fancy way of saying it works before rather than after. Okay. Now we're going to look at another recessive phenotype of 2 genes working in the same pathway that won't give you the ratio 9:3:4, but tend to give you a ratio of 9:7. And how do you get that? Okay, so you have a precursor that's white, you have an intermediate that's white, and you have a product that's blue. If you have a pathway that looks like this, white being converted to white being converted to blue, and you set up some crosses, so you have a enzyme that's converting white to white, and then you have an enzyme that's converting white to blue. So we're going to do a self-cross, big A, little a, big B, little b, heterozygous individual, cross to a heterozygous individual. What's the color of this heterozygous individual going to be? It's going to be blue, correct? So you're going to do a blue heterozygous cross to a blue heterozygous. And you're going to ask, what do you get at the end of this cross? Being heterozygous for 2 genes, you're going to produce 4 different gametes, big A, little a, etc., etc. Sorry, big A, little b, big A, big B, etc., etc. Now, let's fertilize these gametes together, and you're going to get big A, big B, big A, big B, big A, big B, big A, big B, big A, big B. So they're all going to be blue because you have the A enzyme and the B enzyme. So then you're going to get a big A, little b, big A, little b, that's going to be white. Big A, big B, that's blue. Big A, little b, that's white. Big A, big B, that's blue. Big A, big B, that's blue. And now here we have a little a, big B. And this little a, big B will also be white. It will be accumulating precursor. It won't accumulate the intermediate, it will accumulate the precursor. But the precursor is still white. So you won't see it, it will appear white. Again, little a, big B will be white. Big A, big B will be blue. Little a, big A, little b will be white. White and white. So when you get a ratio that is 9 blues and 7 whites, again, it's recessive epistasis, but it's recessive epistasis such that the intermediate and the precursor have the same color or the same phenotype. There's no fluctuation in those phenotypes. So you can't distinguish the steps. So you, at this point, you can't tell whether enzyme A is converting precursor to intermediate or intermediate to product. It can be either one. Okay? You just cannot figure that out because both of them are going to be white. All you can say is that there are 2 different genes producing 2 different enzymes that are taking a precursor, converting it to an intermediate, converting it to a product. So you can figure out that there are 2 genes involved in this pathway, but you don't know the order in which they function because the precursor and the intermediate have the same phenotype. This is also a form of recessive epistasis. And in this example, you're going to basically get 9 blues and 7 whites. A different ratio. That 7 is basically a combination of the 3 and the 4 that you saw previously. So an altered ratio of 9331 is an hallmark of epistasis. So when you get an altered 9331 ratio, that's an indication that there are 2 genes, but those 2 genes are working in the same pathway. Okay. So this is mutations that are recessive. Okay, now we're going to look at mutations that are dominant. So let's look at color in plants, in the squash. Okay. So let's, gene Y determines the color of the pigment. Okay. It's an enzyme that will basically determine whether the color is yellow or green. If it's wild type Y, you get yellow color. If it's recessive Y, you will get green. Gene W determines the production of pigment. This is actually producing a protein that represses this gene. It kind of turns off this gene. The presence of the protein W will basically prevent this gene from making pigment. Or will prevent the color production. So when you're big W, you get no pigment being made. When you're little w, the pigment will be made, but the color of the pigment will depend on whether you're big Y or little y. So the dominant allele of W will block the function of the Y gene. Yes? Does that make sense? When you have the W, big W, it's producing a protein that's basically going to turn off this production of pigment gene. And when you turn off the production of that pigment gene, you're going to basically be white. And then when you have the mutant version, little w, that allows this gene to make pigment. And it will be either, if you're big Y, you'll be yellow, if you're little y, you'll be green. So, when you take a heterozygous white squash and cross it to itself, this squash is going to be white because it's got big W. And it's not going to allow any of this pigment to be made. So it's going to be white. So a white squash crossed to a white squash, you're going to get a ratio that's going to look like this. So you're basically going to get 12 that are going to be white because they're big W, 3 that are going to be yellow, and 1 that are going to be green. So a ratio that goes 12:1 is basically also an epistasis ratio, but it's a little bit more complicated. It's not a linear pathway ratio, but it's a ratio where one gene affects the production or the expression of a second gene. And this is a dominant epistasis because it's the dominant W gene that is affecting the other gene. So this is dominant epistasis. The dominant allele of one gene, big W, hides the effects of both alleles of the second gene. It's hiding whether you're big Y or little y doesn't matter. Big W is hiding the effect of the Y allele, or the Y gene. And so that's epistasis when you have a dominant allele. Okay. Now, there is a second form of a dominant epistasis effect. And let me see if I have that example. If I don't have that example, let me just mention. So right now, the big W is hiding the Y gene. And it doesn't matter if the Y gene is big Y or little y. There is another form of dominant epistasis where only big W will only affect big Y. It will not affect little y. All right? That's also dominant epistasis. And when you have something that works like that, the ratio is 13:3. Instead of 12:3:1, it's 13:3, because this 12 and this 1 will combine together. Okay, so keep in mind that it's 9:3:3:1 ratio, alterations is epistasis. Then you have to remember the ratios that lead to an alteration. 9:3:4, 9:7, 12:3:1, 13:3. 4 different alterations that you have to remember in this class. All right? And based on those 4 alterations, you can figure out whether it's recessive epistasis or dominant epistasis. And if it is recessive epistasis, you can draw the pathway. If it's dominant epistasis, you can't draw the pathway. You just have to basically know the rules and apply the rules. And I will have to give you the rules in this class, because usually those rules are figured out using other experiments. So the fact that W is a repressor of gene Y, you can't figure out just by doing the cross. You have to do additional experiments to figure out what W is doing. Okay? Okay. Oh yeah, I do show you dominant epistasis 13:3. So dominant epistasis 13:3, you see that in basically color in chickens. Okay. So colored feathers are due to the dominant gene big C. White feathers are due to the recessive gene little c. Okay. So big C is color. Big C is color. Little c is white. Okay. But then there's another gene called I, which inhibits the expression of the color. But little I will allow expression of the color. So it's only big I that will inhibit expression of color. Little I will not have that issue. And so when you do this kind of an experiment, only birds that are little I will be colored. Everything else will basically be. And when you're doing experiments like this, you need to be told these rules. And so when I give you problems like this, I will tell you the rules. I will lay out for dominant epistasis, I will lay out the rules. I will say big C does this, little c does this, big I does this, little I does that. And then I'll say, okay, I'm going to take a true breeding X cross to a true breeding Y. I'm going to do a self-cross. What are the ratios to it we expect? So you apply the rules. You draw out a Punnett square with the 4 different classes of gametes. You fill them all out, and then you apply the rules. And depending on that, you'll be able to figure out the color. A little bit complicated, but it's not that complicated. You just have to remember the classic Mendelian Punnett square for 2 different genes. We're only going to work with 2 different genes for something like this, for epistasis experiments. And you're basically going to figure this out. All right. Yes?
Could you figure out what's happening without the Punnett square?
You can. You can do a branch diagram. You come to the same answer. You just have to figure out the different 4 classes that you get. You write those out, and then you basically apply the rule after the fact. So you can write out big A, big B, big A dash, big B dash, big A dash, little b, little b, little a, big B dash, little a, little b, little b, little 4 top classes, correct? You can write those out, and then you apply the rules that I've given. What is big A and big B going to be? What is big A and little b going to be, etc., etc. All right. So you can absolutely do it by branch diagram, no problem. In the book, they always give you these kinds of Punnett squares. It's easy to visualize, and that's why I'm sticking with them, but you don't have to worry. Okay. So this is basically a 13:3 ratio where you get a change in color. Okay. So the key point to keep in mind when I've been putting this here all the way through is that a simple altered 9:3:3:1 ratio of a heterozygous to a heterozygous is an indication of epistasis. And once you get that, you can memorize those 4 classes of ratios, and you'll be able to answer the question very well. So this is the table that you have in your book. This is the one of the few times where I want you to memorize something because it will turn out to be easiest. Okay? And that's the altered ratio. So 9:3:3:1, which becomes a 9:3:4 ratio, a 9:7 ratio, 12:3:1 ratio, and a 13:3 ratio. 4 different ratios. Those numbers should immediately tell you it's epistasis and what kind of epistasis, whether it's recessive epistasis, whether it's dominant epistasis, and if it's recessive epistasis, what is the part involved. All right. So now we're going to look at a ratio that changes the phenotypes, but the ratio still stays 9:3:3:1. So let's take a dihybrid code, or dihybrid cross for 2 genes, A and B, for color of the feed. Okay? Or sorry, lentils. So you can have a brown lentil cross to a gray lentil. A brown lentil is big A, little b, little b. A gray lentil is little a, big B, big B. It's going to produce 2 kinds of gametes, big A, little b, and little a, big B. They'll fertilize. In the F1, all of the gametes are going to be dark brown, or green, or brown, in this case, whatever. They're given different colors. Okay? You can choose dark brown over here, let's call them. Now you do a self-cross. So when you do a self-cross, you're going to get 9 that are dark brown, big A and big B. 3 that are tan because they're big A and little b. 3 that are gray because they're little a and big B. And then 1 that's green because it's little a, little b. This is basically just the fact that each one of these genes affects the other gene in terms of the production of color. And you get a ratio that is 9:3:3:1, but the ratio, there will be 4 different phenotypes at the end of this experiment. 2 genes are controlling coat color, but you need to be told the rules for those two genes. And you don't know how they function. They function in more complicated ways than a simple pathway. And so you have to know the rules of each color, and then you can basically apply. I will basically tell you that if your big A and big B are going to be brown, if your big A and little b are going to be tan or light brown, if your little a and big B are going to be gray, and your little a and little b are going to be green. And once you know those rules, then you just apply them to the Punnett square, and you get the ratios. So in a lot of these epistasis experiments, the rules have to be figured out by independent experiments. And in this class, of course, you can't do the independent experiments, so you'll be told what those independent experiments are. Same thing happens here. Color in peppers. So, gene 1, big R. Big R gives you red. If you're little r, you get yellow peppers. Gene 2, big Y. There is absence of chlorophyll. It's not green. Little y, you produce chlorophyll and you're green. And so you get 4 different possible ratios. You get big R and big Y, so you're red. The reason you're red is the big R will produce red color, and your big Y, so there's no chlorophyll, so there's no green pigment. And because there's no green pigment, it's just white, you're basically going to be red color. Big R and little y is going to be brown because big R will produce red, little y will produce green, and red plus green gives you kind of brown or orange peppers or whatever you want to call it. Little r and big Y will give you yellow because little r produces yellow pigment, and big Y will produce Y, which is no chlorophyll, so basically you'll be yellow. And then little r, little y will be basically green because little r is yellow, and then you'll also produce green pigment, and the green masks the yellow, and so you basically look green. This is 9331 ratio as well that you will see. And then you get more complicated pathways. So we've been talking about the white gene, correct? You guys are kind of familiar with the white gene. It's on the X chromosome. Morgan used it in his experiments on sex chromosomes. The white gene basically is a transporter of pigment. It's present in the facets of the eye, and whatever pigment is made in the cells, it transports them into the facets of the eye, and therefore you see. So when you have a normal white gene, it transports whatever pigment is made and expresses it in the facets of the eye. And the pathway is basically, there is one pathway that takes a precursor and converts it to a brown pigment, and there is another pathway that takes a white precursor and converts it into vermilion. And when you transport both brown and vermilion into the eye facet, the eye looks red. So there are 2 parallel pathways that basically merge. And there are 3 different genes in this example. You have a gene that takes this white precursor, converts it to a brown pigment. It's the gene B+. Drosophila geneticists love to make things complicated. Okay? They label the mutant based on the phenotype. So just imagine, if you have a mutation in this pathway here, what's going to be made? Which pigment is going to be made? Just vermilion, correct? And that vermilion will be transported into the facet of the eye, and it will look vermilion. So a mutation in this enzyme in this pathway gives you vermilion colored eyes, so they label this enzyme enzyme B for vermilion. Okay, so it gets a little complicated. It's just nomenclature. I could have called it A and B, or B and C, or whatever. All right? So don't freak out about it. But a mutation here will make the eye look vermilion. A mutation here in this pathway will make the eye look brown. Mutation here will make the eye look white, because none of the pigments that are being made will get transported into the facets of the eye, and the eye will look white. And that's your classic white mutant. Now, what happens if you have a mutation in both of these enzymes here? What color would the eye be? It would also be white. So you can get white if you are B minus, B minus, or you can get white if you are W minus, or you can get white if you are B minus, B minus, W minus. And you can see how the relationships will work. So if I give you this pathway, I can give you an experiment where I can take a fly that's one or the other mutant cross to another one and ask you. Once I give you the rule or the pathway, then you can apply the rule and figure out what the phenotypes will be. Right? So it's just pathways. It's basically pathways. Think of pathways as basically roads or paths leading to some destination. And then a mutant basically blocks that one of those pathways. And you just ask, if I block it here, what am I going to find? I'm going to find whatever is upstream or whatever can bypass it. And you basically figure it out. It's like driving. You basically, if you have a roadblock on one street, is it possible to bypass it? And if you do, what are you going to see along the way? And you might be able to see vermilion or you might not. It depends. So sort of analogous, not exactly analogous, but kind of sort of analogous. But in all of these epistasis experiments, usually we're going to give you all these details. And we're going to ask you what are the ratios when you do cross A, take a fly like A mutant cross to a B mutant. So you just apply the rules that you're being provided and you'll be able to answer it. And in the practice homework, I've given you examples of this. So practice through them. I've given you several examples of this. Even the homework in the textbook, there are some examples like this of epistasis. So practice. This is again, same thing we just went through. Okay. So we've been talking about 2 genes. We'll gradually make a transition to 3 genes affecting phenotypes. And that was the eye color in flies. Now we're going to look at multiple genes. So for most complex diseases, most diseases like diabetes or Alzheimer's or Parkinson's, it's not just one gene that mutated gives you a phenotype. There are a few examples like that. Cystic fibrosis is one, hemophilia is another, thalassemia. Those are diseases where a single mutation in a single gene gives you a mutant phenotype in humans. But most complex human diseases involve multiple genes in a pathway. And they're not even in a linear pathway. They may be merging together. So how do we study this? How do we try and study this? Right? And what are the effects? So, many human traits, height, skin color, etc., and diseases are determined by multiple genes. More than one gene affect your height. It's not just one gene that affects height. Same thing with color of your hair, your skin, your eyes. There are multiple genes that are involved. You just saw that with the fly. There were 3 genes that are involved in the eye color of a fly. And similarly, there are multiple genes that affect eye color in humans. And so multiple genes affect certain traits that you are studying. Okay? And not very often, these are additive effects. Each trait, each gene makes some amount of a material. When you have 2, you are slightly better. When you have 3, you're slightly more better. When you have all 4, you're great. And so these are known as additive effects of multiple genes. So height of plants. Let's imagine, this is just an example, that the height of a plant is controlled by 4 genes. Okay. And I give you the rules. Genes A, B, and C will contribute 3 centimeters to the height of a plant. This is just a made-up example, but I'm giving you an example to explain something. And these alleles that are not recessive Yeah, alleles that are recessive do not contribute to the height. It's only when you're dominant, so when you're big A or big B or big C, you will add 3 centimeters to the height of a plant. And this is a diploid plant. In addition, you have the 4th gene, gene L. It's always found in the dominant condition, and it gives you a fixed length of 40 centimeters. That's the rule I've given you. So if you're dominant for A, you get 3 centimeters extra. If you're dominant for single A allele, 3 centimeters extra. If you're dominant for single B allele, the 3 centimeters extra.